Welcome back. We're going to be plowing into chapter 3. This is a really, really important chapter to the point where I want to make sure that we get this material and then we'll start plowing into the laboratories. So, this particular chapter, as far as the slides are be concerned, I broke it down into 4 parts, and that way I don't bore you too long at any one time. So let's go and dive right on in. So we're going to be looking at macromolecules and how important they are in terms of making up all living things. We're going to talk about the major groups of macromolecules, the categories of some of the components of a macromolecule, the carbohydrates. We're going to be looking at lipids and the shape and function of proteins, and then we'll finally get into just sort of an introduction to DNA and RNA. We'll cover a little bit more about that when we get into biotechnology. So, food is made up of organic molecules. Of course, that's carbon containing molecules, carbohydrates, lipids, proteins, and nucleic acids. And macromolecules are sort of a mixture or could be a repetitive of any of those listed above. So we're going to talk about that. So a macromolecule are large organic molecules made up of building blocks or subunits. 4 types of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. And we'll show an example found in living organisms. The way this amazing chemistry is arranged, linking carbons and hydrogens, all of the octet rules and things that we talked about in the previous chapters coming to bear now. And when you see the structures, you shouldn't be alarmed, because everything we've talked about in terms of satisfying, like carbon needing 4 more bonds, we have 1, 2, 3, 4. So they're sharing those bonds, a double bond with an oxygen. That shouldn't bother you. This group is called a carboxyl group with a 2 double bonded oxygen and an OH group bound to a carbon. That is a very common arrangement, usually at the end of a chain. And we see it perhaps in smaller chains, nitrogen containing. This is an amino acid, sort of. This is glycine. And here's a circular type structure. It's usually held together, in this case, with an oxygen. And just so I'll mention it many times, this is glucose. And the numbering convention for it is start with the oxygen and always go clockwise: carbon 1, 2, 3, 4, 5, and 6. So it's a 6 carbon system. And the arrangements of the hydroxyl groups and the hydrogens are important, because that's really what dictates if this hydroxyl group went below this circular or this plane, then it would be something else. But we're not going to worry about that right now. So these are just some examples of carbon-based arrangements, and we're going to be seeing these a lot. So Carb Hype, the carbohydrate hype. Hardly a day goes by we watch magazine, TV commercials, YouTube commercials for whatever, sport drinks, Gatorade are filled with carbs. And we're going to talk about carbon loading, carbo loading, as far as coaches will do that to promote better athletic performance. Instead of, let's say, eating a group of candy bars or something like that, or something that's just laden with sugar, you have something that has carbohydrates. It takes the body a little bit more to break it down, and it gives you a more smoother release of sugar throughout the day. And so you eat these power bars and things to get started, and then over the time the sugar is released. As we know, what goes up goes down, and if you eat a lot of sugar, your sugar levels will go up, but it also goes down, and actually goes lower than it was originally before you ate the sugar. So you'd be like worn out, tired. So that's what coaches and stuff are trying to prevent. So carbohydrates include macromolecules that function as fuel. They're primary fuels, and they're they form the structure of all of cells in all organisms, the CHO. So here is a typical carbohydrate linked together sugars. The bonds are really important. There's two types that we really worry about, the alpha and beta, and I'm going to talk about that later. But really what we're talking about is harvesting chemical bond energy, the bonds between uh, the various atoms, and that's what fuels life. This is what drives life. And I have a bunch of animations and things that we're going to go into during this course that'll convince you that really what we're doing is running a bunch of nanomachines. So it's quite impressive what's going on here. And so for right now, let's get the basics. So the carbohydrates are just mostly carbon, hydrogen. You can see some examples up here, and oxygen. Sometimes they contain atoms of other elements, but they must have the CHO. And carbohydrates generally have approximately the same number of ratio, like H2O. For example, best known carbohydrate is glucose. The composition is C6H12O6. And so you can see that this ratio, the carbon, hydrogen, oxygen, 6 to 12 to 6, is pretty common. And it's just an observation. It's nothing much more than recognizing that. So just to break it down, carbohydrates, and they're composed of monosaccharides, simple sugars, and we've got glucose, fructose, galactose sort of drawn out in a ball structure. And this carbon to hydrogen store energy, and we're going to talk about what that energy is in a minute. So they're macromolecules that most consumers are somewhat familiar. To lose weight, you want a low carb diet. Athletes often carb load, as we've already mentioned, to make sure you get somewhat of a standard level in your blood instead of going less than what you started with, which you feel really tired. But here's the linear version of glucose. I had shown you the circular portion with the oxygen being sort of the go around, but we can see the oxygen here. But here's the various carbons as a chain. So there's glucose, galactose, and fructose. And if you look between galactose and glucose, really we're looking and comparing. If your sharp eyes can detect the differences, it's which side of that chain the hydroxyl group and the hydrogens exist. They're pretty much the same, but this one, glucose, is flipped, and when it's flipped like that, that becomes galactose. So if it's a circular, it would be above the plane. If it's not, it's below the plane. But in a in a linear fashion, you can see clearly the difference. Fructose, the double bonded carbon, occurs on the number 2 carbon in the chain, and so as a result you have a cyclic that occurs with 5 carbons instead of 6. This one hangs out above the molecule. So anyhow, we'll get used to these structures as we use them more and more. Carbohydrates are primary fuel for running all of our cellular machinery, and it is in fact machinery, and I will convince you of that by the end of the semester. You can quote me on that. Carbohydrates form much of the structure of the cells of all life forms, and it's the easiest way to store energy. A chemical bond energy, of course, is the currency of life, and the chemical bond energy is what does the work. It changes shapes of things so that various events can occur. Carbohydrates contain carbon, hydrogen, and oxygen, and they have the same sort of ratio as the water unit. So it would be CH2O, and if you ramp it up, you'll see the ratios pretty much stay the same. Simplest carbohydrates, including monosaccharides, the simple sugars that contain 3 to 6 carbon atoms. As the chemical bonds of carbohydrates are broken down, Other more stable bonds are formed, and a great deal of energy is released. And it's that energy, that chemical bond energy, is what drives life. And I'm going to show you exactly how that happens later on. So glucose provides that energy for body cells. So energy needed, molecular bonds are broken, and the fuel for cellular energy. And what this does is it affects the change in shape of a molecule. But if you don't follow me right now, that's okay. Energy not needed, it's stored. And glycogen stored for short term and then long term as fat. So I think we are aware of that. So carbohydrates, the most important of living organisms, glucose. The simple sugar is naturally found in most fruits. We eat table sugar called sucrose, but that's really a disaccharide. And starchy carbohydrates found in bread, potatoes are converted into glucose by your digestive system. So glucose circulates your blood in a concentration of about 0.1%. So that's 0.1 grams per 100 mLs, and that's a lot, really, if you think about it. But we have to drive all of our life energy, so circulating glucose is called blood sugar, and it has one of three fates. So there's glucose, and you can see 1, 2, 3, 4, 5, and then the 6 carbon up here, and the hydrogen. The oxygen is what kind of holds the ring together. Okay, so now we look at a chemical bond energy. Let's talk about that. So we have the attraction between hydrogen protons, the hydrogen bonding. We've already talked about that, pairing of electrons. And if we pull these apart, now they want to be stable, and it takes a fair amount of energy to do that. So we have to add into the mix about 436 kilojoules per mole. And per mole is also represented as mole to the -1, which means it's 1 over the mole. So they usually do that if you're writing it in a sentence, but kilojoules per mole is what usually, don't let that confuse you. And the delta H means the change in the amount of heat. And this is a chemist shorthand. And kilojoules we're going to talk about as a measurement of energy or heat released in the system. It's usually how it affects a certain amount of water to bring it up to a 1 degree or something like that. So we'll talk more about it, but all chemical reactions involved in the breaking and making of bonds. So, and when we break a bond, then they make products, and in the process of doing that, we harvest energy. And the electrostatic forces of attractions between atoms and ions, to break the bond, you must give the element energy in order to shake it up enough. You know, if you were to add energy, just start shaking something, eventually it will break apart. And that's just a simple way to kind of look at it. So bond energy. Now we're going to talk about this energy later on in terms of being created nor destroyed. We're going to talk about enthalpy or bond energy. The bond enthalpy, or the amount of energy between two hydrogen bonds, is about 436 kilojoules per mole. And I already mentioned it takes about that much to break every hydrogen to hydrogen bond or hydrogen binding. Bond energy also tells us how strong the bond is. The higher the bond energy, the stronger the bond. Double bonds have much higher bond energy, enthalpy or energy than single bonds. So, not to be technical on you, I just throw in some vocabulary at you because we're going to be using these terms later on. And the more you use it, the more it becomes more natural to you. If you're confused by that right now, don't worry about it. Energy always is required to break a bond. Energy is released when a bond is made. So the exact value, if I use the carbon to hydrogen bond as a good standard, it takes roughly 100 kilocalories of energy to break. And 100 kilocalories per mole is sort of a standard, what we use for heating water. But if you want to convert kilocalories, everyone knows, how many calories a piece of food contains, and roughly the amount of heat it takes to heat water is kind of the standard that they use. But I had already mentioned this kilojoule is another. The physicists will use this term a lot And so there's a website if you want to use that. You can just copy the link out of my slide set here, and you can enter the kilojoules per mole, or you can add how many kilocalories per mole. And I added 100 and then allowed the calculation to give me 418.7 roughly kilojoules per mole. And so a kilojoule is defined as 1 kilocalorie, 1,000 thermal gram calories per 1 mole of substance. So 1,000 calories is the amount of energy or heat needed to increase the temperature of 1 gram of water, or 1 ml of water, it's the same thing, or cubic centimeter, by 1 degree C. And so that is, if you can kind of think of, everyone's heated water before, and if you had the amount of energy for 1 gram of or 1 ml of water for 1 degree C, you can kind of roughly know where 1,000 calories will kind of get you. So relate that best you can when you look at how many calories let's say a candy bar consumes, and in order to burn that off, so it doesn't go to fat, you got to run for a certain amount of time, and your body will heat up and all that. And what you're doing is you're utilizing the energy in chemical bond energy to cause muscle contractions and various other things. And about 40% of it just is lost as heat, not lost, but transformed into heat. And we'll talk about that later. But anyhow, that's the big picture. And so we can link the monocarbohydrates together in these long chains. We have starch, we've got glycogen, we're going to talk about those, cellulose. If you notice, the bond angles are a little bit weird. This structure is flipped, even though the bonds look the same, but they flip the chemical structure. This is actually a beta link. Now, these are alpha, and beta links are different. We can't break these down. Chitin, the same sort of thing. They're beta. So anyhow, very strong bonds for these, because we can't technically break them down with our enzymes. They have to have other types of animals and creatures that break them down, with bacteria really doing the work. So if we add water to one of the molecules in the link and a little bit of energy, we break the two, and the water is resolved. So we're causing hydrolysis, or dehydrolysis is the removal of water. Adding water is hydrolysis, and you can see there's the OH and H group. It resolves that bond and some energy to do that, and we have to add somewhere about 28 to 34 kilojoules per mole. Gives free energy. Now, this is complicated. This gets into the chemistry of things, and I'm going to bring it up. I'm not going to run from it. It's really just a matter of getting familiar with Gibbs free energy, and it's sort of counterintuitive, but the free energy is the amount of energy in negative signs of a delta G is minus 28 to 34, and it's the temperature and other factors that relate to how much energy there is. And so instead of we would think of it giving off energy as we break, it may utilize energy, and we represent that as Gibbs free energy. I'm not going to have you worry too much about it, but I'm going to still use the terms so that the learning process is there. Reactions that have a negative G, or Gibbs free energy, release free energy, and called exergonic reactions. And it's interesting because you would think that if it's giving off energy it would be a plus symbol, but no, it's the exergonic. It's the same sort of argument that a physicist will give you when you put your hand on a cold table. It feels cold. Well, what's actually happening, it feels cold to you because your hand is providing energy or heat to the table. And so the atoms and all that are transferring energy from your hand to the table, and your nerves see it. Your sensory perception is that it feels cold. And it's because it's taking energy. Anyhow, that's sort of the relationship that we see with Gibbs free. Exergonic means it's exiting. In Latin, exit means to leave or to go. So exiting the system. So if you think of it, exergonic means that energy is leaving. And a negative G means that the reactions or initial state have more free energy than the products or the final state. Exergonic reactions are also called spontaneous. So it's the same idea as I was mentioning with a hand on a cold tabletop. And it's the energy exiting your hand. You can think of it that way. At reactions where the positive gives free, or in other words, the values are positive, it requires the input. So the idea between the two is that we're releasing energy and we see it as a negative number, and if we're requiring energy, we see it as a positive number. Anyhow, that'll help you if you ever decide to take a chemistry class or move into other areas of biology. Water weight. Now, I have to tell a story on myself. When my wife had my first child, she gained weight, of course, and had a child. And I gained weight too because I was worried about how I was going to pay for it. That's a common thing. Well, when she loses the weight and has the child, I didn't lose the weight. So I needed to get on a diet and lose some of that weight. And I had to cut back on eating and all that, but the first thing, guys usually lose weight faster than women. It's just kind of a known thing, and it always upset some of my friends who were female that said, "Oh my gosh, how do you lose so much weight?" And they didn't. It just took them longer. And I find out later, we'll talk about it, that vitamin D kind of helps balance that out. You can actually lose the weight like men if you have a sufficient amount of vitamin D. But don't do anything unless you talk to your doctor first, of course. That's always going to be the case. But because a pound of glycogen has about 4 pounds of water molecules bound to a dieter's lose water weight during the first days of a diet, because of course when you're resolving and breaking bonds, of course you're using water to do that, and as a result you're consuming it, so your body's going to start to weigh less. And anyhow, so that's kind of the deal with that, and it's common, but if you hold true, in about 2 weeks you're going to be losing some serious weight, and it happens, and so you can do it. Glycogen plays a role in rapid weight loss because you reduce your caloric intake and your body's burning more calories than you're consuming, and that way you don't need to store it. So that's pretty standard. When I step on the scale, it says 1 at a time, please, but I just thought I'd add that. So in summary, glucose is the most important carbohydrate in living organisms. Glucose in the blood can be used as an energy source, which would have to be there. It can be stored as glycogen in muscles and liver for later use. It can be converted to fat. A lot of times if you consume certain fatty foods like milk and things, sometimes you get what's called a fatty liver, and that's because the liver, of course, converts. It's the chemistry lab of your body, and it actually starts to build fat tissue on the liver itself, and that's not a good thing. So complex carbohydrates, in contrast to simple sugars, contain more than one sugar unit. Two simple sugars can be joined together into a disaccharide, such as sucrose, table, or lactose. We're going to talk about both of those. Many simple sugars sometimes have 10,000 or joined together, resulting it's called a polysaccharide, and it depends on the sugars and things that you're binding. But it's really a time-release storage, and of course, if you want to utilize this in the long term, it will do that. Example of structural material is polysaccharide cellulose, the primary component of plant cell walls. Cellulose we can't break down, but it's a plant's way of storing energy. Glucose can be hydrolyzed to produce ATP or stored in animal polysaccharide glycogen. So the glucose bond energy is actually useful in producing the universal energy form of currency, I guess you would say, ATP, adenosine triphosphate. And the ATP is accepted in more places than MasterCard, I can tell you that, in a human body. Glucose monomers can be generated from the breakdown of other disaccharides, such as lactose. So that's the summary on that. So starch in plants, the primary energy storage in complex carbohydrate is called starch. It's found in roots and tissues. It consists of linked glucose, which can be broken down in an alpha configuration. And grains such as barley, wheat, rye are high in starch content, such as corn and rice, both of which is about 70% starch. Glycogen that stores energy in your muscles and liver also is complex. We refer to this as animal starch, and it's usually more branched, but it's still composed of alpha links. So these alpha linked starches are important. Now, the beta linked ones I was referring to before, like in cellulose, we can't break these down, and we don't have the enzymes to do it. We'll talk about what enzymes are. But you can see the beta, and we have to flip the structure around in order to make it look like this one up here. This one's upside down, and it gives you that beta-like link. And bacteria are able to break it down, and I'm going to give some examples. So starch is just one of those starchy foods. Pancreas, we have enzymes like amylase that breaks down the fat or the various food types, and then breaks that down such that it's absorbed into the bloodstream and broken into glucose. And then we have hormones, which is part of the endocrine system, that insulin helps cells take the sugar in, or glucagon that helps make resistance to that sugar, and so that we keep a good balance, how much sugar we should be having in our blood and how much this energy form that our body cells can take. Now, the enzymes convert substances into different molecules as products. So that's what enzymes do. They reduce the amount of energy it takes. They can be reused, and we're going to talk more about that. Where the endocrine hormones are chemical messengers that are secreted directly into the blood, and then goes to their particular places to exert the function. So the hormones are very specific, as enzymes are, but hormones are typically utilized and used up in that reaction. Pancreas serves two functions to break it down. Insulin lowers blood glucose levels by increasing glycogen synthesis and storage in the liver. Glucagon increases blood glucose level by limiting the synthesis of glycogen in the tissue. So there are some interesting relationships talking about insulin resistance and diabetes. And if you're interested in these sorts of things, there's a plethora of information you can find about that. It seems to be very common these days to have glucose resistance and diabetes type 2. It seems to be the new epidemic. So complex carbs, simple sugars, many disaccharides, polysaccharides, just means di means two, poly means multiple, are important cell sources of fuel for our cells. Unlike simple sugars, disaccharides and polysaccharides must undergo some processing. So that delays a little bit. Although the energy is stored, we can get to it. It just takes a little bit of time to get there. And sucrose, common table sugar, is composed of monosaccharides glucose and fructose linked together. To use sucrose, the body must first break down the glucose, the links between the two, to get glucose and fructose free. And then once they do, then we can utilize the chemical energy as a result. So sucrose, being the common table sugar, has glucose and fructose, as I mentioned, and then breaking this bond here to release that. So it takes a little bit of energy, but we get a whole lot more energy out of the chemical bonds that are within the glucose to make ATP. And again, we'll be talking about that. Complex carbohydrates, just sort of as a review, if we break the bonds, or in this case, building the bonds to make the long chains like starch, and then we later, through digestion and breaking these bonds, to get and harvest the energy back. So this is sort of the ability to store that chemical bond energy for later use. Now, I had mentioned before lactose, and this is sugar, And when we break lactose, we get galactose and glucose. Again, it's fructose and glucose if you break down sucrose, but there's an enzyme called lactase that breaks down lactose into galactose and glucose. Now, you may or may not be aware that some are lactase insensitive. Or lactose insensitive because they don't have a functional form of lactase and the enzyme to break it down. And so, and that's a problem because you get indigestion and unpleasant results, and just by drinking milk or something like that. So, lactase is a digestive enzyme that breaks down lactose. And there's a lot of factors, and we're going to look at this in one of the labs, things that affect the enzyme from working properly. And we'll look at this. When the breakdown ceases in the human body or insufficient ineffective lactase results as lactose intolerant. Now, women that usually have a child will experience temporary lactose intolerance for a period of time because some of the hormones that are produced actually block the sites for the enzyme. And what I mean by that is typically you have a perfect glove and hand fit for the enzyme. Let's say this is lactose, and it was usually referred to generically as a substrate binding into the active site of an enzyme. When it fits into that particular shape, the enzyme will change its shape to some degree, put a stress on the bond between the two, in this case, the galactose and glucose, and then you get the products galactose and glucose released. And what this does is greatly reduces the amount of energy it takes to do this, and the enzyme's free to go and do it again, and that sort of thing. But in lactose intolerance and after pregnancy, one of the hormones invariantly will bind into the active site just because it has a similar structure, and for a while you cannot break down lactose, and that's sometimes a problem. So, but you get it back. Enzymes reduce energy required to break down the compound. So without an enzyme, the amount of energy starts to increase. You can see this in this graph with a red line, and that's activation energy, the amount of energy it takes to break the galactose and glucose. But with an enzyme, the enzyme puts enough stress on that bond by putting physical at the molecular level stress on that bond, it's easier to break it. And as a result, less energy is required, and so it greatly enhances the rate at which we can make these products, and also the amount of energy is greatly reduced. You can see it's almost 60-40, so about 60% less energy to break that bond, roughly. So let's talk about, I had mentioned carbon loading, carbo loading early on in the presentation, and this really is better spoken with the short-term versus long-term type of energy that our body has. Of course, what Coach doesn't want you to do is eat cupcakes and sugary foods that have simple sugars, and you'll get this huge burst of energy, which is great, but the problem is it drops, and this graph is kind of not totally accurate. It actually goes down below what your initial sugar level was and then balances out, but that's okay. Long-term energy that's with complex carbohydrates, in other words, bound up glucose molecules and the like, you can see the body is accessing and breaking it down, but it takes a while for it to be delivered, and as a result you get this sort of smoothing out effect, and this is what coaches will look for, and you'll get a better effect performance wise over a longer period of time, instead of the crash that we might experience down here where you're just dog tired. Here you still have energy, maybe not as much as the highest part, but still pretty good. So that's what carbo loading is all about. And a specific example has been well illustrated, and this is great for folks that are diabetic as well. Peanut butter is a great runner's food, and peanut butter and jelly sandwich is a great diabetic food. But the idea is the carbohydrate and the sugars are bound up as a complex type, and the peanut butter has to be broken down, and that's the loading effect. And over time, and you'll get the benefit of it as you go on, and you can read more about this if you're interested in this sort of thing, and most folks are. So multiple simple carbohydrates sometimes link together into more complex carbohydrates. And the types of complex carbohydrates include starch, a primary form of energy in plants, and glycogen, in a form of energy storage in animals. So these are important. We're going to come back to these, but again, every chapter that we have kind of builds on itself. So the chemistry that you've learned is now coming to focus. Next time, we'll be talking about more details as we get into more of the chemistry of some of these other molecules. And I hope you enjoyed this part, and I'll see you next time on part 2 of chapter 3.